Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about different types of vectors which are used in recombinant DNA technology. So what is vector? Vector is basically act as a vehicle for transporting the foreign DNA into the host cell. Okay, for the purpose of cloning and expression. So there are different types of vectors. Vectors in DNA recombinant technology are DNA molecules that carry foreign genetic material into the host cell, where it can be replicated and expressed. Common vectors includes plasmid, bacteriophage, cosmid, phasmid, artificial chromosomes. Each type serves as a vehicle for gene cloning and multiplication, with plasmids being the most frequently used due to their ease of manipulation, okay, and replication. So plasmid is a type of vector which is most commonly used, okay, but there are different types of vectors which we will discuss in our video. So what are the characteristic features of the vectors? At first, it should be capable of replicating autonomously. The size of an ideal vector should also be small enough for it to be incorporated into the host genome. The small size of vector also enables it to incorporate a large sized insert for the transport. So it should have a small size. Vectors should be easy to isolate and purify, as these need to be recovered and reused for multiple purposes. Next, many vectors also require unique restriction enzyme recognition sites that enable the insertion of vector DNA in presence of specific restriction enzymes. So they should have unique restriction enzyme recognition sites. Then introduction of vector into host cell should be easy. In the case of gene transfer process, it is important that vector is capable of integrating itself or recombinant DNA into genome of host cell. It is important that the introduction of recombinant DNA into the vector does not affect the replication cycle of the vector. So these are some characteristic features which are to be present in an ideal vector. Next, types of vectors. At first, cloning vector. So cloning vectors are vectors that can be capable of replicating autonomously and thus are used for replication of recombinant DNA. We have to insert our desired piece of DNA inside that vector, then it should replicate autonomously by itself within the host cell. Cloning vectors are responsible for determining of which host cells are appropriate for replication of that particular DNA segment. Cloning vectors are further different types that are defined by different features unique to each type of vector. So cloning vectors are used according to our goal, our purpose, according to the host cell, okay. Next, one of the most important cloning vector is plasmid vector. So what is plasmid? It is present mostly in mostly in bacteria, and these are small extra chromosomal circular DNA molecule which are capable of replicating autonomously within the host cell, and these are naturally occurring vector. These are termed as workhorse cloning vector in recombinant DNA technology. Plasmids are widely used as vector in 3 domain of life. That means it can be used for prokaryotes and archaebacteria and the eukaryotes. However, these are frequently used in bacteria and yeast. The most important feature of plasmid that makes them one of the best vector is their small size. The small size of plasmid facilitates the separation of recombinant DNA from the host genomic DNA. So it can carry large piece of DNA segment because their size is too small. And one of the most commonly used plasmid vector is PBR322 and PUC19. Okay. Next is cosmid vector. Cosmid is a type of cloning vector which is a hybrid molecule that combines the features of both bacterial plasmid and bacteriophage, enabling to clone large DNA fragment. It contains plasmid DNA with an ORI, origin of replication, within the bacterial cells and cos site from lambda phage, that means bacteriophage, which is necessary for packaging of DNA into phage capsid for delivery into bacterial host, because bacteriophage can attack and insert the DNA into the bacterial cell. What are the key components and features of cosmid vector? One is presence of cos site. What is cos site? A DNA segment from the lambda phase that facilitates the packaging of cosmid DNA into phage capsid. Next is ORI or origin of replication, a DNA sequence from plasmid that allows the cosmid to replicate independently within the bacterial host cell. Then antibiotic resistant gene, a selectable marker such as ampicillin resistance gene to identify bacterial cells that have successfully taken up the cosmid vector. Then it should have a multiple cloning site, a region containing restriction enzyme sites where foreign DNA can be inserted. Okay. So these are some key features or components of a cosmid vector, which is a hybrid vector, hybrid of bacterial plasmid and bacteriophage site. Bacteriophage, the part of bacteriophage DNA, cos site. Okay. Next, bacteriophage vectors. Bacteriophage also act as a vector, and bacteriophage are virus that only infect the bacteria and transform them efficiently while carrying large DNA insert. Bacteriophage or phage have higher transformation efficiency, which increase the chances of recovering a clone containing recombinant DNA segment. The most important feature of phage is packaging system, which enables the incorporation of large eukaryotic gene and their regulatory element. The use of this bacteriophage also facilitates the isolation of large quantities of DNA, which can be used for analysis of the insert. So, the most common bacteriophage vectors are M13 phase, lambda phase and P1 phase. Okay. Next, bacterial artificial chromosome. These are engineered DNA molecule mainly used to clone DNA segment in bacterial cell. These consist of bacteria derived F factor replication origin, which enables the propagation of large DNA fragment in a supercoiled circular form. Bacterial artificial chromosome can carry larger DNA insert as compared to plasmid or phage vectors. So these are more efficient than plasmid vector and phage vector. Next, yeast artificial chromosome vector. These are also engineered DNA molecule which are used to clone DNA insert within yeast cell, particularly Saccharomyces cerevisiae. Okay. These are developed in order to clone large sequence of DNA, so to increase the efficiency of the process. It can clone up to 500 kb of DNA, which is higher than most of the traditional cloning vector. Next is human artificial chromosome vector. So these are extra chromosomal DNA fragment that act as new chromosome within the human cell. The use of this human artificial chromosome HAC has increased with advances in genetic engineering, as it helps to overcome problems commonly associated with traditional vector system. These are exist as single copy episomes without integration into host chromosomes, allowing them for long term stable maintenance. Okay. Next is viral vectors. Viral vectors are one of the most effective means of gene transfer to modify host cell or tissue and manipulate them to express different types of genes. The concept of using viruses as vector arose from the fact that viruses are very effective in transducing their own genetic information into the host cell. The process is called as transduction. Okay. During viral transduction, the non-essential viral genes can be replaced with foreign DNA sequence or our desired piece of DNA sequence of therapeutic interest or important gene in order to produce recombinant viral vectors. When these viral gene enter inside the host cell, it will express and produce our desired product. Next, expression vector. Expression vectors are vectors that enable the expression of cloned gene in order to determine successful cloning process. So these are expression vectors. Expression vector is a DNA molecule, typically a plasmid or can be a virus, designed to introduce a gene to the host cell and direct the synthesis of protein encoded by the gene. So it should contain the elements like promoters, terminators to control the gene expression, often include antibiotic resistance genes for selection, then affinity tags for protein purification. So these are essential tools in molecular biology for producing large amount of specific protein inside various host organisms like bacteria, yeast, mammalian cell. What are the components? It should have origin of replication, promoter for initiate transcription, coding sequence for a desired protein to express, then terminator to end the transcription, one selectable marker to identify and select the host cell. Next, translation control sequence, initiation site, stop codon to ensure proper protein synthesis. Okay, so these are key features of expression vector. Next is subtle vector. These are engineered plasmids which are designed to function and replicate in two different host organisms, most commonly in prokaryote and eukaryote like yeast and mammalian cell. They achieve this by incorporating multiple origin of replication, selectable markers, one set for each host. Okay. Next, secretion vector. These are the vectors which are specialized expression vector that expresses the cloned genes in order to produce proteins at locations other than the cytoplasm. The transport of protein product from cell is achieved by the fusion of insert DNA with nucleotide sequence encoding peptide of an easily secreted protein. The use of secretion vectors has many advantages like higher yield, a simple purification process, improved protein stability. So these are designed for more than one type of prokaryotes as well as eukaryotes, including mammals. The most commonly associated problem with incorporation of a protein of eukaryotic origin into prokaryotic host is overexpression of the protein. This problem can be solved by the use of secretion vector that elevate the formation of inclusion body. These have replaced cloning vector in the processes focusing on the production of protein and the expression of eukaryotic DNA fragment. So these are special type of specialized expression vector that contains DNA sequence encoding signal peptide which directs the recombinant protein product to be secreted out of the cell rather than remaining in the cytoplasm. So this facilitates high yield of protein, simplifies purification process, it improves protein stability by reducing the formation of inclusion body. Okay, so secretion vectors can be used in various biotechnology applications such as producing therapeutic proteins like insulin and developing vaccines. Okay. So with this I am concluding my lecture video here. In this video I have discussed about different types of vectors which can be used to transport DNA or desired piece of DNA into the host cell. In my next video I will discuss about some commonly used vectors, some important plasmid vector like PBR322, PUC vector. So we will discuss some examples of vectors. So if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel to get more important videos like this. Thank you.